So, good afternoon, everyone. On behalf of Indian Society of Clinical Research East Zone and Indian Pharmacological Society West Bengal chapter, I am welcoming you all in this grand academic event that is on advances in drug development. So, we have galaxy of faculties today with us. We are really fortunate to have them today with us. First of all, may I request Professor Tapon Kumar Mondol, President of Indian Pharmacological Society West Bengal chapter, to address us. Over to Professor Tapon Kumar Mondol.
Hello. Hello, Sambo.
We can hear you, sir.
Hello, Sambo.
We can hear you, sir.
Okay. Namaskar. Heartiest welcome to all of you joining the online webinar that is on advances in drug development organized by Indian Society of Clinical Research East Zone and Indian Pharmacological Society West Bengal from Inovocare e-Academy today. We know the drug development is an important step in the field of pharmacology. There are many reasons why new drugs are important, such as new diseases, development of drug resistance, and increasing understanding of health conditions, allowing treatment of previously untreatable conditions. And stages of drug development are discovery, product characterization, formulation, delivery, packaging development, disposition kinetics, preclinical toxicology testing, and IND application, bioanalytical testing, and also clinical trial, and involving a large amount of fund, around $1 billion with a time period of 10 to 15 years. So today, we have organized such type of symposium. Our learned speaker will speak on the latest development for new drug development involving less amount of expenditure with a short duration. I will welcome to Dr. Sanis Davis, President ISCR, who will deliver keynote address. Dr. Subrajyoti Bhaumik, Chair, ISCR Eastern Zone. Heartfelt welcome to the speakers, Dr. Anup Rawal, Consultant Clinical and Cancer Genetics at Fortis Hospital, Mumbai, and Associate Director, Medical Genomics Division at SRL Diagnostic Lab, Mumbai. Dr. Sandhya Selvarajan, Head, Clinical Pharmacology, JIPMAR, Pondicherry. I also welcome the chairpersons, Dr. Rusha Pingali, HOD, Clinical Pharmacology, Nizam's Institute of Medical Science, Dr. Somid Malhotra, HOD, Clinical Pharmacology, PGI MER, Chandigarh, Dr. Urmila Thate, formerly HOD, Clinical Pharmacology, KEM Hospital, and SET GS Medical College, Mumbai, and also Dr. Soibal Mukherjee, formerly VP and Head Asia Medical Science Group. Once again, welcome to all the participants, member of IPS West Bengal branch, ISCR Eastern Zone, and also Dr. Sambo, the General Secretary of IPS West Bengal branch and key person to organize such type of symposium. Wish the program a grand success. Namaskar. Over to Dr. Sambo.
Thank you, President sir. Thank you for your welcome address. Now we have to move forwards towards the keynote address, and we are really fortunate to have Dr. Sanis Davis, who is a very senior clinical pharmacologist and also the President of Indian Society of Clinical Research. Welcome you, sir. Please have the keynote address. Over to Dr. Sanis Davis, sir.
Thank you, Shambho. And if you may allow me to share my screen. Can you confirm that you can see my screen, Shambho?
Yes, sir, we can see.
Thank you. So at the outset, thank you to ISCR East Zone chapter, as well as the West Bengal Indian Association of Pharmacology for the opportunity, and also to Inovocare for, you could say, the collaboration. For those of you who are not aware about ISCR, it stands for Indian Society for Clinical Research. It's a member-driven organization enhancing patient outcomes through clinical research. ISCR actually started in 2005, and you will later on hear one of our stalwarts, Dr. Shoibal Mukherjee, who was instrumental in setting this up several years back, or almost more than now close to 2 decades ago, where there was a felt need that there is no association of stakeholders who are involved in clinical research. So it's a organization or a society which actually looks at the discipline of drug development as well as clinical research and clinical development and everything that falls into that space. Now, of course, you have several societies or associations which look at very small parts of clinical research or clinical development. But ISCR actually tries to encompass all the different specialities within clinical research or clinical drug development. ISCR actually works through councils and chapters. These are all smaller, you could say, task forces or groups which actually focus on a particular area in the research spectrum or continuum. And they have their own, you could say, mission and vision. So the overall vision of ISCR as an organization, and we welcome all of you to be part of it if you're not already part of this, is to ensure that you have an ecosystem for clinical research to be built in India. And as many of you who are researchers as well as budding researchers would like to actually do research within India, it's important for you to actually see that there is a vibrant and a thriving ecosystem which can actually use your skills as well as also give you the benefits of being part of the research community. Given that research within India has several diverse stakeholders, including the government as well as academic institutions, industry, patient advocacy groups, etc., you need everyone to pull together if the ecosystem actually works well for everyone. So ISCR as an organization looks at several different wider, you could say, tracks. Some of which is to ensure that the research landscape in India actually is working well for everyone. We have a lot of advocacy initiatives that we drive with policymakers and regulators and others in the health ministry, etc., to ensure that we have a robust ecosystem for research. We also ensure that there is adequate awareness both within the public as well as in the media on the importance of research. And in hindsight, when you look at the COVID pandemic, there is one thing which actually the COVID pandemic as well as the fallout from the pandemic actually did for research. It is the awareness that many lay people actually have about research, about vaccine drug development, etc., etc. So COVID has been a big boost to actually clinical research in our country. There's also a lot of capacity building educational programs which ISCR run. There is also an academic consortium for clinical research which has mentorship programs for young researchers. There is annual awards for research that anyone does. It need not be pharmaceutical industry research. It's for research that is held in academia. So that happens every year. We also have other training programs as well as lifetime achievements, etc. ISCR also has a peer-reviewed index journal, which is the official publication of the society. It's published quarterly. It is well quoted. Dr. Arun Bhatt actually helps it. Many of you who are listening, who might be either a clinician or a clinical pharmacologist or from the academia, would usually ask, what is in it for me if I join a society like ISCR? And to put that in perspective, both medical education, pharmacy education, etc. in India traditionally does not have formal teaching or training in doing clinical research. It's more like it's like a byproduct of some of the courses that you take, etc. But there is no formal training that anyone actually gets when you're in the medical school or in a pharmacy college, etc. And many advancements in academia are currently linked to whether you have publications or you are doing research, etc. So it's a good thing that is coming up as part of the NMC guidelines and others. And every medical society, whether you take endocrinology, whether you take medical genetics, whether you take gastroenterology, any medical society has research as a pillar apart from that specialty's own skill development in many ways. And many clinicians, especially young clinicians or young professionals who would like to do research in the midst of their active clinical practice, do not usually have easy access to gaining expertise or skill sets in doing that research. And that you could say lack of that skill sets or that expertise prevents people from actually getting into research in a big way when there is no dearth of clinical material in India, and that's such a waste for the nation. The environment for doing clinical research has significantly changed post 2019, both for a regulatory research as well as for academic research, and that's a great thing that has happened for India. A society like ISCR can be that bridge where researchers can easily find answers and support, both for embarking into academic research as well as for clinical trials, if it is done by the academic industry. And the nation needs more of these committed researchers if we have to actually move forward into drug development and research. So who can join ISCR? Anyone who's actually listening to this, whether you are a student, whether you are a academician, you're a clinician, or even if you're in the industry or the government, etc., you can be part of ISCR. It can help in many ways, not just you, but also the wider society in a big way. So with that said, I probably had a couple of thoughts with regards to advances in clinical development. And I feel that as researchers or people who are in the drug development discovery industry, there are probably three things which are still the pillars for drug development or discovery. And many a times we all, you could say, equate drug discovery as drug discovery and drug development as the implementation of all this, right? And the three things that I have is, one, of course, where does it start from? It all starts from what we call as the label of the drug that ultimately comes out in the market, the target product profile, or the TPP as they say, and of course, linking all this together is the clinical development plan. As researchers, many would actually not think about the end product, but if you have to bring a drug to the society, a useful drug to the society, then as researchers, you need to think about what would this drug ultimately end up as, and that's the label of the drug. And from that label actually comes the promotional claims, and from there, I think you have to think about what should you do as developing the drug in order to get to that label or to that promotional claim, etc. So while people would equate the label on the drug to be the commercial equivalent of what you could say people in the sales and marketing division of the drug company or anywhere else would think about, actually the label is the most important product that the company's research wing actually produces, because this is what gives the drug the approval, also conveys how this drug is going to be used, and of course it describes all the efficacy and the safety of that particular product. So any researcher who wants to go in a big way in drug discovery development also has to think about what would be the ultimate use of that drug, and thinking about the label is an important part. Linking the label to the clinical development work is what is the target product profile. This is actually initially started out as a format for discussion between the sponsor and the regulatory agency. But over a period of time, this has become like what we say as the Janam Kundli for the compound, right? As to how information is going to change what you had as an initial thought and ultimately what it will become, which is what you'll see as the marketed product. So it's a dynamic text which is used to actually iterate on how the drug is actually moving from preclinical studies all the way up into the clinical studies and into the market. So the TPP will have all these discussions about what should be in the label, what all studies will you do to actually support that label, and ultimately what kind of discussions will you have at what time points with the regulatory agency, etc. And this is an example of how you actually would put in, you could say, the efficacy part for maybe a new drug that you're developing for a condition like asthma, where you already have drugs which are approved in this indication. There could be drugs which are looking at being developed for that same indication. And there could be your drug, which is where you have to look at what is the minimum that you would want in your drug from an efficacy perspective and a safety perspective, etc. So it's a you could say a document which will look at the iterative process. Now, to bring this TPP into what you have to do on a daily basis is what is called as the clinical development plan. And that's a vision which is transformed into distinct steps, what we call as clinical studies, each with its own milestones and deliverables. So if you look at a clinical development plan, this is for maybe developing a new SGLT2 inhibitor. We all know that these drugs are going to be used in patients with diabetes, but is that what you want? Or are you looking at a drug which has other kind of actions also? So you might have to study that drug in whether it decreases hypertension in these kind of patients. Will it be useful in patients with renal issues? Will it also be useful in type 1 diabetes, etc., etc.? So you start with the label, you move into the TPP or the target product profile, and then you ultimately end up with the clinical development plan. This is the whole continuum or the three pillars that you're needing for your drug development. So to end my, you could say short, you could say thoughts about this. Why do we think this is all important, drug development, right? The strategic vision of the nation is now moving into Vigyan or Anusandhan, as you could say the government is stating. That would actually translate at the ground level that while India's burden of the non-communicable diseases are going to overtake communicable diseases in the near-term future, we can't let, you could say, drug development for some of the ever persistent communicable diseases like malaria, dengue, and TB to be left to somebody else to do. Nobody will do it for us. We'll have to do it for our own country. And drug discovery and development cannot actually start, you could say, suddenly, right? While COVID was a very exceptional example where everyone pulled together and there was a lot of ground support for everything, whether it was the government or academic investigators or the industry to do, it may not be true for all these kind of diseases. So drug discovery and development needs an ecosystem to thrive. And that ecosystem cannot just happen in the industry or in academia or in the government by working in silos. There has to be everyone working together. And today's session, like for example, the 2-hour, 3-hour session that we have is part of that overall capability and the capacity building. And this cannot be restricted to only a few centers of excellence. This discipline of drug development and discovery has to continue to grow. And we need all the stakeholders to pull together, the government, the institutions, the researchers, industry, patients, to make drug development as a discipline to be more successful. So thank you so much and wish you a happy listening.
Thank you very much, Dr. Sanis Davis, for your wonderful keynote address. And you had perfectly actually set the learning platform for all of us. And as you had mentioned repeatedly about COVID-19, and there is not like that COVID-19 is, yes, it is of course a bane for this civilization, but also it raised something like some boon also with this COVID-19 pandemic, and most importantly this type of online learning sessions, and we can have lots of very good teachers from globes in this type of platform. Along with that, another important awareness that was raised about the drug development. Initially, it was almost about among the basic scientists, pharmacologists, clinical pharmacologists, and some clinical clinicians who are engaged in phase 3 clinical trials. They actually are interested about this type of drug development strategies. But after this COVID-19 pandemic, the vaccines development and drug development, we have some curiosity in our minds that what actually is going on with this drug development. So once again, thank you, Dr. Sanish Davis, sir. We have to start the technical session now, and for the first session, that is on bioinformatics applications in drug discovery and development. We have two very noted clinical pharmacologists as chairpersons. We have Professor Shomir Malhotra, sir, who is the professor and head at Department of Clinical Pharmacology at PGI MER Chandigarh. Welcome you, sir. And also we have Professor Usha Pingali, madam. Madam is the head at Department of Clinical Pharmacology at Nizam's Institute of Medical Science. Welcome you, madam. And we have today's speaker, Dr. Anup Raul. Dr. Anup Raul has completed his DM in medical genetics from Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. And he is now the Associate Director of Medical Genomics Division at SRL Diagnostic Lab, Mumbai. He also looks after the reproductive and germline Sanger and NGS analysis and reporting different NIPT, PGD, PGS and NGS panels and exomes. He is also working as consultant clinical and cancer genetist at Fortis Hospital, Mulund, Mumbai. So now may I request Professor Shomir Malhotra sir and Professor Usha Pingali madam to start today's session. Over to Professor Shomir Malhotra sir and Usha Pingali madam.
So good morning everyone, good afternoon rather. I thank ISCR for giving me this opportunity along with the ITS West Bengal branch and especially Shambho. He has been pretty active and always interacting with us. And of course, my co-chair, Dr. Samir Malhotra. We all know that bioinformatics provides a wide range of drug-related databases and softwares, which can be used for various purposes related to drug designing and drug development. And we also know that bioinformatic analysis not only accelerates the target drug selection, identification, refinement, but it also helps in characterization of drug-related side effects and also predict drug resistance, now, which are the in thing nowadays, especially in relation to oncology. And high throughput data, we know that data from pharmacogenomics, genomics, epigenetics, and proteomics. Now, all this has made significant contribution to mechanism-based drug discovery and drug repurposing. And to speak to us on this very important and interesting topic, bioinformatics applications in drug discovery and drug development, we have none other than Dr. Anup Raul. I welcome you, Dr. Anup. And before I hand over to you, I hand over to Dr. Samir Malhotra, who is my co-chair, to say a few words. Over to you, Samir.
Okay, hello, Usha. Dr. Sanish, hello. Shambhu, hello. I think if Dr. Shoaibal has joined, hello to him also. Anyway, good afternoon. I think Dr. Usha has made a perfect introduction, And without waiting anymore, let us go straight to the topic. Dr. Anup, please.
Hello, am I audible?
Yes.
Yeah, you're audible. Yeah.
First of all, let me thank the Indian Society of Pharmacology and Indian Society of Clinical Research for giving me this opportunity to speak in the esteemed symposium. And special thanks to Dr. Shambhu, have made a very new and kind friend now in West Bengal. So bioinformatics as such is a naive branch, and it is something like when we do MBBS and after that we want to specialize further, what else? So it is something like a science which is evolving slowly. Much of it has gone into the sequencing aspect, next generation sequencing labs, molecular genetics labs that have come up, and lots of application and qualified intelligentsia is put in place there, and they're practicing. But apart from that, there are a lot many aspects, or I'll say multidimensional facets of bioinformatics, which are relatively unexplored. Yeah. So this was on the cover of Time magazine way back in 2011, and what it was postulated was by 2045 humans will become immortal. And look at the computing power, the explosion that they were looking at. Like by 2015, it was supposed that the computing power of a computer will be that of equal to a single mice brain. By 2023, it will be like it has already, we have exceeded that, and prior to 2023, we have already achieved what the computing power of a single brain, human brain. And by 2045, what they estimate, like we'll have computing power which will be equivalent to entire human race in a single computer. So that is what the technical or explosion or evolution we are looking at. So today the topic is bioinformatics. The focus will be on its applications in drug development, drug designing. I'll be covering a few topics, some interesting articles I'll be sharing. And my focus particularly will be on pharmacogenomics, how it is important from the drug development point of view, and also the clinical aspects. So as it was really, we are all aware, like drug development is a huge and tedious process involving a lot of time and a lot of money. And this is the facts that were shared in 2021 for top 5 pharma companies in US for the fund allotment that they do in the routine. So R&D is about 16%, and this is really exploding. So throughout the journey of drug development, there are certain bottlenecks, certain obstacles that are there, something like false discoveries, the preclinical studies, the strategies, they are not very effective. Then through the conventional studies that we design, we are able to study only a handful of targets. And then most important thing is RCTs, which is the definitive step for validation. It comes at the end of the pipeline. Before that, it is not there. So these are certain loopholes. Interestingly, a lot of impetus and money or research has been going on in oncology, field of oncology. Lot many drugs are being studied. Lot many drugs are being repurposed, and if you compare the number of drugs that are being churned out in the field of oncology and compare it with other specialties, definitely there's a huge contrast that is to be seen. And it was by Danisher, it was previously pointed out, like, yes, if you look at the evolution in drug development, or just to take a comparison in vaccines. So historically, if we look at the years it was taken, or decades it was taken to come out with one successful vaccine targeting one particular disease condition. So years together. But if you see 2020 COVID, in almost 1 to 1.5 years, we have come out with a vaccine. So what was the strategy that was behind it? That needs to be understood, because same can be applied for drug development and drug designing. So bioinformatics is a multidisciplinary study where we integrate molecular biology and information technology. And anyone with a basic knowledge of biology and computer sciences can get into this field, right? There is no limit, or there is no barrier to it. It is all based upon your interest. And again, within bioinformatics, there are certain areas which need to be focused depending upon your interest: pharmacogenomics, data mining, protein modeling, and so on. And again, with the advent of certain technological aspects like artificial intelligence, machine learning, and deep learning, so this is increasing the versatility and applicability of bioinformatics in a lot of sense, in a lot of ways. And same applied to the conventional drug discovery. This can bring a lot of revolution kind of thing, because you can end up cutting costs. You can do a lot of hypothetical studies or computational studies, and then based on that knowledge you can plan your actual clinical studies for a particular molecule. And this can also save on the time, workforce, and money, of course. So genomics, a lot of us are a bit skeptical when we talk of genetics or genomics, and the fault is the basically curriculum which never covered this aspect. Genetics has evolved much more in the research field than in the academics in medical like MBBS curriculum. So just to brush up before we move on. So human body made up of millions and trillions of cells, each cell having a nucleus having a chromosome. And chromosome is nothing but a supercoiled structure DNA, which is one single strand of DNA divided in this 23 chromosomes. And this chromosome hosts what we call the genes. So certain segments of this DNA, which are protein coding, what we call as genes. And this is the DNA structure of DNA, double helical structure made up of nucleotide base pairs arranged in a particular manner, which are bound together by hydrogen ion bonds. And each gene codes for a particular protein, which has a particular function in the human body. So some genes will be coding for receptors, some for enzymes, some for connective tissue, and so on. So when we talk of genomics, so this is the spectrum of omics. So genomics, transcriptomics, proteomics, and metabolomics. So this is the whole concept of omics. It turns, or it is focused on the central dogma in genetics. So central dogma is like the transfer of information from DNA to RNA, proteins, and to the further metabolites that happen. So at each step, this is definitely governed by a certain set of mechanisms that go on to have a role play in the at the molecular level in the cells. So DNA with transcription giving rise to RNA, then RNA translating into proteins, and so on. And each step is what we call, like if you're studying study or focusing on the DNA, then it is the genomics or genotype studies that you're doing. Something to do with DNA modification, acetylation, methylation, and so on, then that is the epigenomics. If you're studying the expression pattern of those genes on the DNA, so that is you're studying the transcriptomics, and so on. So why this understanding this concept is important? Because if any abnormality is there at the level of DNA, it manifests at various levels, and it gives rise to a certain set of clinical picture, what we call as phenotype. Now, understanding this is important because this will help us when we are looking at the disease. What is the disease condition? What is the molecular abnormality underlying that disorder? And by that molecular abnormality, we want to figure, okay, is it targetable? Like, is there any drug which is already there, or a similar molecule which can be selected? Or maybe a molecule is there which is different, but mechanism may be similar and it can be repurposed. So things like this. So all this information is gradually being cataloged in various databases. So there are databases for human genotype, the HGMD, and so on. For phenotype and genotype, there is CleanVar. So there are many databases that are there, which are open access, which can give us the information that we want. So practically bioinformatics, it complements for basic up and to combine or just to bridge across the basic upstream and downstream analysis. So at the molecular level, what is happening, which genes are mutated, upregulated, downregulated, which pathways are affected, what is the effect on the cellular function on at the level of proteomics, what is happening at the level of metabolomics. All this information is collected, and based on the abnormality that is there, what is the disease in picture that we're talking of, and what is the clinical phenotype. So such databases are created, and there is an interlinkage that happens when we do a bioinformatics analysis to pick up particular mutation which may be associated with more than one disorder, and vice versa. So this is very important in bioinformatics. So basically, the basic bioinformatics person is something like a wet lab person. So when you ask a wet lab person, he may be working on PCR, or he may be working in a biochemistry lab, or he may be doing analysis on mass spec. So different things. So similarly, bioinformatics is a wide area. One could focus on particular sub specialty of it. So just doing next generation sequencing, data analysis and reporting in a NGS lab, or could be doing a comparative phylogenetic study, or could be studying or doing a research on population genomics, or in the pharma industry on drug development, and so on. So when we talk of drug development, so there is a drug development cycle that goes on through various phases, preclinical and clinical, just to summarize. So it all starts with drug discovery. So to have a lead molecule and to identify targets where this molecule is going to work. So when we have any data pertaining to, and so if you're looking at an antibiotic, so if I have a data for that particular organism where I have the hotspot of mutations that can be targetable, then I can look at the molecules which are available, which target those. If over time resistance have developed and I need to develop a new molecule, I look at the mechanism why this resistance is happening and how I can overcome this resistance at the molecular level. So is there any existing molecule which can bypass this? So all this information can be obtained. So there are websites like Cake Pathway, which does analysis on this fronts and gives us data. So this is one website that I'm quoting, but there are many other databases where you can just give the information. You can, pull in the information that you want based on the organism that you want to study and based on the drugs or targeted molecules that you want to look at. So this becomes important in the first stage of the preclinical drug development cycle. And then there are ways and means based on this identified criteria, like you can do analysis based on this information in lab. So you can develop models like knock-in or knockout models to look at to study the molecules that you want to have a look. And again, all such information can be looked into the databases, public databases. And most importantly is whenever such studies are done, such information is readily available. So if for any particular mutation in a particular gene, a study is already done that shows that, okay, this particular molecule is not going to work on that hotspot. So this helps other clinicians to avoid doing a replication of a study which is going to be futile in terms of manpower and money. And then there are various aspects. So bioinformatics is just one aspect which is looking in so for drug discovery from multidimensional things, like what is the intended molecule that I want to study, what is the disorder that I'm looking at, And based on these two aspects, what is the underlying molecular mechanism that is involved? What is the drug metabolism that is going to happen, and how it is going to influence by the transcriptomics of that particular organism. So therein comes the role of systems biology. So this is all intricate process where, based on using bioinformatics as a tool and gathering information from various aspects and looking at the target molecule, lead molecule, and the drug target interaction, we can speculate which molecules are going to work or which may or may not work. And we can also design the drugs accordingly, and we can avoid unnecessarily pulling in those molecules which we know that are not going to work in the study. Then same thing based on the bioinformatics data, sequence alignment for, so if one particular hotspot region, like suppose for HIV, there is one particular gene CCR5 which helps the virus in internalization in the T cells. So individuals who have mutation for or a variant for this particular gene, the receptor is rendered non-functional, so that HIV virus internalization is not possible, right? So based on such information, if we are going to target this particular gene or this particular receptor on the cells of in a clinical study model or in an in vitro lab model, so same information can be withdrawn. So if I want to do a study on cell line, what will be the difference of interpretation or the mechanism in the cell line vis-a-vis the human model? So this type of studies can be done based on the computational modeling. Similarly, homology modeling for determining the protein models, then how the protein or ligands are going to interact. So this can be done by molecular docking method. And again, molecular dynamics to set standard for comprehend trajectory of lead as well as its interaction. And most importantly, to look at the pharmacokinetics and dynamics aspect and the adverse drug reaction aspect of such molecules. So identifying the drugs, then using prediction models, system biology, and artificial intelligence in drug designing based on whatever information is at hand. And most importantly is, can we develop a hypothesis on computational analysis and try to figure on those molecules which will probably be successful or pass through this stage of the drug development. Most importantly, in preclinical stages, this pharmacodynamic and pharmacokinetic interactions, the ADME. So this is very well studied in oncology drugs, because apart from the drug interaction, environmental factors which have a role to play in terms of the clinical effect or therapeutic effect benefit of the drug, but at the same time there are certain individuals, a set of individuals who have adverse drug reactions which could be severe, and there could be a cohort of patients where they neither get adverse reactions but neither do they get therapeutic benefit. And therein, in such cohort, there is importance of underlying genetic factors which govern the drug metabolism and the overall clinical outcome. So many enzymes have been studied which govern the phase 1 and phase 2 metabolism. Even this information is helpful when we are designing newer drugs. So what will be the influence in phase 1 and phase 2? Which enzymes are going to interact and modulate the efficacy of the drug? And what is the underlying genotype which is governing the functioning of the drug? So very very common example is of Irinotecan and UGT1A1. So the active metabolite of Irinotecan, SN38, is conjugated by UGT1A1 and excreted from the body. And SN38 is very highly toxic. So this Irinotecan is used for treating cancer patients, colorectal cancer. So if conjugation of capacity of liver enzyme UGT1 is decreased because of any pathogenic variant, there will be accumulation of SN38 in the body, and it will give rise to toxicity, severe neutropenia, severe diarrhea, and there will be catastrophic condition. So in almost 1 to 1.5% of the cases or patients who lack this conjugation capacity, they have very severe life-threatening adverse reaction for irinotecan. So similarly, at the level of DNA, what are the variations which could be at the level of single nucleotide or a number of it, some tandem repeats, some deletion duplications, how this governs the function of the gene, whether the functioning of the gene to produce a particular protein product is increased, amplified, or decreased, nullified. That governs the action of that gene on the functioning of that enzyme. So on two fronts, on pharmacokinetics, the ADME factors, where the transporters are involved, where plasma protein binding is involved, and metabolism of the drug is affected. And on the pharmacodynamic front, receptors, where internalization of the drug molecule is hindered. Same thing with ion channels, enzyme, where either the prodrug is broken down into active drug, or active drug is rendered inactive by an enzyme, and again immunomolecules which govern the pharmacodynamics. So just to give an example, so warfarin and VKRC1, CYP2C9 and VKRC1, they govern the function of warfarin, and practically across OPDs, clinics, patients who are on warfarin, if there are 100 patients on warfarin, roughly 1 to 2% have an adverse genotype which render them to a life-threatening bleed, even with the minimal dosage of warfarin. And again, these patients are susceptible or prone to drug-drug interaction, adverse drug, rendering them again susceptible to bleeding disorders. So on pharmacodynamics fronts, a very good example is RYR1, where dantrolene, malignant hyperthermia results when there is exposure to volatile anesthetics. And then this immune-related, so HLA type, immune-mediated type 4 hypersensitivity reactions, particularly modulated to HLA genes. And the classical example is abacavir in HIV patients, allopurinol in rheumatoid arthritis patients, and so on, in epilepsy, carbamazepine. So this also figures in adverse drug reactions, susceptible patients. Next is the toxicological aspect. So this is important because most of the drugs fail in preclinical phase at this level. And again, if there is a prior knowledge, like studies shows that a particular molecule is very prone to develop to give rise to adverse drug reactions, then in drug designing front, can certain qualities of the drugs be altered, or the structure can be altered where you can render it and curb the adverse drug reactions. So such computational studies can give results and help us to plan for better drug designing in such patients, in such studies. Then data landscaping or data mining. So on particular disease condition for a particular molecular genotype, so has there any studies been done? Like for example, the very good example is in HIV CCR5 gene. So many studies are being done and documented or cataloged in one particular database. If you can Google for CCR5 database, which shows like, okay, which molecules are going to work, which molecules can be repurposed, what is the structural similarity. Similarly, researchers keep creating their own databases. Most of the pharma companies have their own databases. Some may not be available or may not be open access. But as and when we have societies like ISCN, so ISCR, sorry. So such databases can be made available to the researchers and it can definitely benefit the community. So toxigenetics or toxigenomics word is used interchangeably. There are two aspects. One is genetic susceptibility to adverse drug reaction due to some alterations within the DNA. So same drug may not cause problem in some individuals, but in particular susceptible individuals, it may give rise to problems. So historically, if we remember fava beans G6PD, so that is one such example. And the omics aspect is very important because what alterations go on the pathway, from the DNA to proteomics, which govern the phenotype or the outcome between outcome of the reaction between the drug and at the molecular level. So that becomes very important. Then comes from preclinical, we come to the clinical aspect. So first time tested in humans. So very limited number of drugs compared to where the pipeline started. Again, if we, with the present databases that is available, if we're targeting a particular disorder, maybe we can precisely bring the number of drugs coming to this stage may be less, but passing further stages, the percentage of drug may increase. So the overall efficacy of this drug cycle, right from the drug discovery up to phase 3, phase 4, the yield of the number of drugs successfully clearing all the stages may increase. So this was what I was talking of. Eventually, when we are talking of any drug for any disorder, there are 4 cohorts of patient that can be divided. So same diagnosis, same prescription, same drug in the same dosage. Few of the patient cohort may just get toxicity, but no benefit. Some will neither get toxicity, but they will also not get therapeutic benefit. In one cohort, the yellow cohort here, the drug is not toxic and not beneficial. And in the green cohort, drug is toxic but beneficial. So based on the genotype, if we can identify beforehand what drugs can be given to which particular patient, because again, this becomes important. The drug may have its therapeutic benefit, but still a certain cohort of patients, due to their peculiar genotype, may not get benefit of the drug, the therapeutic effect. So even that aspect needs to be looked at, what we call it as pharmacogenetics. Coming to phase 2, after phase 1, this drugs, what we see the test or the trials in relevant patient population. And here the limitation is smaller number of patients, and then again common for multiple phase 2 trials, testing various clinical indications, combinations and formulations. Then in phase 3, again compare the trial to current. Now this becomes very important. Is some drug already there? How it supersedes that drug? And here collating information about reproductive toxicology, long-term adverse reactions, if there are any, and in particularly carcinogenicity, it becomes very important. So there are 2 aspects of this. One is from the drugs. So one particular drug, if it is intended to target one particular region and give out a therapeutic benefit, at the same time, is it targeting any molecule elsewhere? Because the bottom line is, in liver, no drug is metabolized 100% by one particular enzyme or one particular pathway. There are some alternate pathways which are metabolizing the same drug. The percentage may be less, maybe less than 10%, 5%, but that could be significant if there are certain mutations which alter this mechanism or metabolism of the drug, giving rise to unwanted effects. Then comes the last aspect, new drug application. So most important is drug repositioning. So previously a drug is known which is already licensed for a particular condition, but on analysis or on investigation we find out, okay, this drug can be used for some other disorder and target some other molecule, or it can alter the phenotype and give some benefit. So this is one very classical case study of Dr. Lucas Wortman, a fellow who was studying on leukemia himself, was diagnosed with ALL in his adulthood, a clinician working on leukemia, had 2 rounds of chemo, relapsed, had a 3rd round, had bone marrow transplant, again a relapse. And that was when a next generation sequencing test was done for him. And they found one particular gene which was upregulated, which was giving rise to the carcinogenesis. And at that time, there was one particular drug that was available which could shut down the upregulation of this gene. But that drug was licensed, available in market and licensed for renal cell carcinoma. So the institute where he was studying, they got a approval and he was given the drug. So that was way back in 2011, 2012, somewhere around that time. And his leukemia was, his leukemia vanished after that therapy. Subsequently, he underwent again stem cell therapy, stem cell implantation, and he's under treatment at present. So this is very interesting, like a rare case, but it gives a good insight, like to what extent bioinformatics can help us. So if proteomics study and transcriptomics study was not done for this one patient, that probable target which was already there and was causing the problem, recurrent relapse, would not have been identified. And at the same time, there was again a drug molecule which was already in the market, which was licensed for renal cell carcinoma, and it was repurposed for ALL. So this is how application wise bioinformatics can help from for repurposing the drugs. So in pharmaceutical industry at clinical stages, right from genomics data analysis, and so what all bioinformatics can help in terms of primary outcomes as well as for following data types which can be generated during the clinical trials. So compilation of data, systemic analysis, again correlating or cross comparing the data across the patient population, across the geographical or ethnic background. So that is one. Then comparison of the data across the disorders or within the systems, then comparison across clinical phenotypes, and most importantly, the testing modalities that are used for and interpretation. So bioinformatics can help on all fronts. The technicalities of application in each field will differ based on the individual expertise, of course. So for drug development on all these aspects, so it is like Microsoft package if you install in your computer. So you have Microsoft Word, you have Excel sheet, you have Microsoft PowerPoint. So based on what you want to work, what you want to present, you pull out that particular software and you utilize the services. So similarly in bioinformatics, depending upon what I want to particularly look at, I will use all these tools and try to achieve what I'm looking at. And just one example in detail, I want to share how in clinics this next generation sequencing is working.
Could you summarize?
Yes.
Because we are running short of time.
Yes, just 2 slides, I guess. So from the initial consult where we obtain the blood sample, we extract the DNA library preparation. The most important part is sequencing, and the next important part is the bioinformatics analysis. So for next generation sequencing, the fulcrum or the important aspect for reporting is the bioinformatics analysis, what we look at. So then looking at some studies which have shown, like bioinformatics research on drug sensitivity prediction. So this study was published in December 2021. So it talked about modeling based anti-cancer drug sensitivity prediction. and some new applications for the data which is already there can be seen. So this basically here they look at the role of drug sensitivity prediction for the oncology drugs. Then there were how the recent advances are shaping bioinformatics. So skin disorders are quite common, a number of them. And now researchers are building a database where you catalog the mutations in those particular respective skin disorders, so as the same can be utilized for developing drugs targeting those mutations. Similarly, multi-targeted curative potential for molecules in cervical cancer. So this was looking at the cervical cancers, the HPV prevalence, and based on the gene expression patterns in the somatic and germline that could influence the overall efficacy of the drugs. So this is how the drug disease entity relationship looks like from bioinformatics point of view. It is multifaceted. We look at the molecule, we look at the molecule gene relationship, how the compound is going to interact, how the phenotype is going to behave, how the omics cascade is going to behave, and how if at all, if there is any scope for repurposing, even that can be looked at. And this is the last slide, most common queries we get. So for Indian population, data is not there. So that is not true. A lot of studies are being done. So for across genotypes, data is being compiled. So as and when this gets updated, same can be utilized. So for various disorders, such information is already being cataloged, and this can be a very good guidance for drug developmental studies going ahead. So just to summarize, this is a unique field. So even pharmacologists can take this field. Very interesting. Just knowledge of biology and computers. It has a lot of potential. So depending upon the interest and what applications you are interested in. So one can go ahead in bioinformatics. Yeah, thank you. So I'll be happy to take any questions.
Yeah.
Dr. Shambhu, we have time for questions.
Yes, sir, with your comments, with you and madam's comments, we can move forwards towards the next session.
Yeah.
Dr. Usha, please.
Yeah, okay. Yeah, thanks, Dr. Anup, for giving us, the overview of the multi-dimensional facets of bioinformatics. Yeah, most of us are pretty new to this field, and you've really given deep insight into it. We know it. Oncology was always the focus of drug development, and with COVID, of course, vaccine also have come into the picture. But one small question which I have to you is, what do you think will be the limitations? Because we know it's extremely useful in drug development and drug discovery, but what problems do you anticipate, or what are your
Yes, ma'am. So
problems?
very nice question, ma'am. So the problem that we anticipate here is presently many courses of bioinformatics are there in India, but they are in institutes which don't have any interaction with medical colleges, academics.
True.
So that is the very important lacuna that I find. And if we can get this aligned with medical universities, then the applications and its utility can be automatically streamlined with our focus.
Sure. Yeah. Yeah, yes, I think that's perfectly said. Unless there is that integration between in academia and the industry doing this sort of research activities, or the labs, basic scientists doing this activity, unless we get into that, perhaps we cannot use it for patient care. The whole purpose of this is to better the patient care, and I think that's very, very useful. And over to you, Dr. Samir. And thank you, Anup. It was a wonderful presentation.
Thank you, ma'am.
Just a couple of comments. Very nice presentation. For a non-pharmacologist to make such a presentation is very nice to see. The lecture started with a quotation about laws of genetics, right? So they
Yes,
are
sir.
like natural laws of physics.
Yes.
Whether we are there or not, they will work. And the time figure that you showed, by 2045 we will be immortal. No, there is a catch, provided our planet is still there. The way climate is going, the predictions are not too good. And as someone has said, the last century belonged to oil, this century belongs to data. So this is bioinformatics is all about this data capture. And one very interesting thing I learned was that we may be able to predict long-term outcomes using bioinformatics. Now, to my mind, this will be exceedingly interesting idea, because carrying out those studies actually is very difficult. If we can just have some data and build models to predict those outcomes, that would be nice. That is all from my side. Welcome, Dr. Sandhya, if she is there, and Dr. Urmila. Hello to both of you. That's all. Thank you. Thank you,
Good
Shambhu.
afternoon, sir. Yeah, I'm there.
Yeah, yeah.
Hello. Yeah.
Yeah,
Hello.
thank you,
Good
Shambhu,
afternoon.
and hi, Sandhya and Madam
Hello,
Urmila.
madam.
Hi.
Yeah.
Yeah, thank you.
Thank you once again, Dr. Anu, for this wonderful deliberation. And we are really thankful to Professor Shomir Malhotra sir and Professor Usharani madam for giving their precious time. And as we now understand, it is an interdisciplinary field, that is bioinformatics. And today we have a perfect mixing of different disciplines. And even we must congratulate Dr. Anup Raul, because also his subject is a beautiful subject, which is totally a translational science, having some clinical parts, having some basic science, and also some translational sciences, just like what the science of clinical pharmacology is. And we have lots of experts today on this field of clinical pharmacology, which is also a totally translational science, having basic science, having some translational aspect, and also a clinical medicine. So with that, we have to move forward towards the next session, next academic session. And for that, we are really privileged to have Professor Urmila Thatte madam as chairperson. Welcome you, madam. Madam, we all know there is no introduction is required for madam. Madam is emeritus professor of clinical pharmacology at St. Cheers Medical College and KEM Hospital, Mumbai. Welcome you, madam, to chair the session. And we have also Dr. Sandhya Selvarajan, madam. Madam is the professor and head at clinical pharmacology at JIPMAR, Pondicherry. Welcome you, madam. Now I request Professor Urmila Thatte, madam, to please.
So Shambhu, I have a PPPI meeting at 2:30, so I will have to leave, and I don't expect Sandhya will finish her talk, so somebody else will have to sum up. So
Okay.
If you could please find someone else who will be able to sum up or end the session. I'm sorry, I'll have to leave before that.
Okay, madam.
Because I think we were running late. So anyway, that meeting has been kept and I've just found out about it. So I have to attend that meeting, the PVPI meeting. It is very welcome, Dr. Sandhya. I mean, we worked together and it is a pleasure to have you as to give this talk. Additional professor and head of the Department of Clinical Pharmacology at JIPMAR. Dr. Sandhya has a DM in clinical pharmacology and has taken more than 8 projects in the last 5 years, and also is the project lead in the Advanced Center of Clinical Trials under the ICMR Intent program. She has 5 RCTs, 11 observational studies, 7 meta-analyses, 3 review articles, and 3 case reports in her publication portfolio. And she's been the member secretary of the Institutional Ethics Committee for Interventional Studies in JIPMAR. since April 1st, 2022. Member Secretary of the Stem Cell Research Center, a research program since 2019. Coordinator of the ADR Monitoring Center, coordinator of the Drug Information Center, member of the Institutional Ethics Committee for the Vector Control Research Program, and member of the IEC of a medical college close by, and assistant editor of the Journal of Pharmacology and Pharmacotherapeutics since 2014. Welcome, Dr. Sandhya, for your
Thank you,
talk.
madam. Yeah, thank you, madam. Thanks for the kind introduction. I will just share my slide, and for better net connectivity, I'll just switch off my video, and again I will join once my talk is over. Is my slide visible?
Yes, madam. Yes.
Yeah.
Yes, it's visible.
Yeah, thank you. First of all, I would like to thank ISCR, E-Zone, and IPS West Bengal chapter for giving me the opportunity to present a talk in this program. I'll be presenting on physiologically based pharmacokinetic modeling in drug developmental research. Before starting the thing, I would like to give you a disclaimer that I have not worked in physiologically based pharmacokinetic modeling, though I have worked on pharmacokinetics. Also, I would like to give one more disclaimer, because of a viral fever which I had recently, my voice is very bad and I'm really sorry about it. I may also cough in between, so I'm sorry about it. So in today's program, I'll be covering the introduction to various pharmacokinetic models, physiologically based pharmacokinetic model, the applications and advantages and limitations, and the summary of this. So to begin with, what is pharmacokinetics? It is simply described as what the body does to the drug. So what the body does to the drug, when the drug is taken, it undergoes absorption, distribution, metabolism, and excretion. But it does not remain the same in everyone. There is lots of inter-individual variations, which results in either less dose or increased dose or change of drug for different people. So how do we individualize the pharmacokinetics, or how do we individualize the treatment? So the application of pharmacokinetic concepts to design individualized dosage regimens to optimize therapeutic response and minimize adverse drug reactions has given rise to pharmacokinetic model. So if you look back at the history, it was Thorsten Theorel, who is considered as the father of pharmacokinetics, to lay the initial stone for pharmacokinetic models. He had published kinetics of distribution of substances administered to the body with extravascular and intravascular modes of administration in 1932. It was the first pharmacokinetic related study where he has drawn by himself so how the drug is distributed in various compartments. But however, there was a long gap before the pharmacokinetic models really came into use. So if you look at the pharmacokinetic models, we have two types of analysis of pharmacokinetics. One is the model approach and the other one is model independent approach. So the first I will say what is model independent approach. Model independent approach does not consider any model, and it does not consider body as having different compartments. It's a non-compartmental analysis, which we are not going to deal today. The other one is model approach, which provides models of data or models of system related information. So what is this models approach? The model approach is basically a mathematical model. Here, the body is represented as a series of compartments that communicate reversibly with each other, and it gives some equations to generate the data. Okay, so what are these compartmental models? We have different types of compartmental model, 1 compartment, 2, 3, etc. So let us just look into it one by one. So 1 compartment model is the simple model. Here, all the components of the body is considered as a homogeneous unit or central compartment. For example, when the drug is given, the drug gets distributed equally in all parts of the body. So the whole body is considered as one single compartment and the drug is equally distributed throughout the body. So this is the model for one compartment. So when the drug is given, it reaches one compartment and from there it is eliminated. So here, if we plot a graph, we will get a straight line. Whereas two compartment model, here we assume that the body consists of two compartments. One, a central compartment, the other, a peripheral compartment. So when the drug is given, first it reaches highly perfused organs. Then after some time, it gets distributed in other parts of the tissues. So when the drug is given, it first reaches the central compartment and then it goes to the tissue compartment and some amount of drug can come back to the central compartment. So what are these K10, K12, K21? So they are nothing but first order rate constant of transfer of drug from central to tissue compartment, tissue to central compartment, and from the central compartment. Similarly, in here, if you plot a graph, we will get a biphasic line. We also have three compartment models where in addition to two compartments, we consider one more deep tissue compartment, which is poorly perfused, for example, fat, plasma proteins, etc. So in addition to central and peripheral compartment, we assume one deep tissue compartment also. So here it will be transfer of the drug from central to peripheral compartment, peripheral to central compartment, central to deep tissue to central compartment, peripheral to deep tissue compartment, and deep tissue to peripheral compartment. So many transfer of drugs will be there. So there are three types of three compartment models known as catenary model, mamillary model, and perfusion model. So the catenary model is the simplest model. It is just like a train. All the compartments are connected to each other. The central compartment is connected to the shallow tissue compartment, which is connected to the deep tissue compartment, and the drug will reversibly come back from deep tissue to shallow tissue and central compartment. But however, this is not a most commonly followed model because the body does not function like this. The drug does not go from one compartment to another compartment and come back in a reversible model. So another model is known as mamillary model. It is also known as the sandwich model, where the central compartment is sandwiched between the shallow tissue compartment and the deep tissue compartment. So whatever drug is given, it reaches the central compartment. From there, it goes to the shallow tissue compartment, deep tissue compartment, and from these compartments, again, drug comes back to the central compartment and it is eliminated. The third model is perfusion model. Here, only the tissues where the drug is going to be available are included in this model. This perfusion model is also known as physiological model or blood flow model. Here, the organs without drug penetration like brain or bone are excluded from the model, and the drug concentration in a particular tissue is predicted by the size, blood flow of those particular organ or tissue. So this perfusion model, the information obtained from one species can be applied to another species. So that is the major advantage of perfusion model. So this perfusion model has given rise to our area of interest today, that is physiologically based pharmacokinetic model. So let us look into Pbpk model. What is a Pbpk model? It's a mathematical model for predicting ADME. It facilitates interspecies transpositions from one mode to another one. It is used in drug development as well as health risk assessment of cosmetics and general chemicals. The Pbpk model integrates drug and system data to simulate pharmacokinetic profile of drug in plasma and tissues. That is, it uses information about the pharmacokinetics of the drug and data about the system, that is the data on physiology of a particular species. Okay, let us look into the Pbpk model. What does it do? It provides a framework for predicting systemic and tissue exposures. In this model, the compartments are defined by the volume, blood flow, and tissue composition, and the information is taken from the species of interest. So here, the information is taken from in silico model, in vitro model, and animal PK model. And then all these informations are used for guiding the optimized ADME properties in humans. So all the information from in silico, in vitro, and animal PK data are used for determining the pharmacokinetic properties in humans. This can be used for predicting first in human dose, as well as to identify successful drug candidates during drug development. So during the process of drug discovery, the Pbpk model can be used for early risk assessment or to predict early first in human dose, toxicokinetic dose, or early formulation assessment based on the in silico, in vitro, and in vivo studies. And even during early development of drug, to predict the first in human dose, single or multiple ascending dose. And the most important application of Pbpk model is in drug-drug interaction to find out the food-drug interaction. Also to predict the pharmacokinetics in other groups of people, like healthy people, patients with liver impairment, liver failure, renal impairment, obese people. So these kind of information can be obtained using these models. And even after the drug is marketed, we can find out the pharmacokinetics of these drugs in special populations using Pbpk model. Okay, so among the physiologically based pharmacokinetic modeling, the simple physiologically based pharmacokinetic model assumes perfusion rate limited distribution within the model. It is almost similar to the perfusion model. So it assumes wherever there is perfusion, the drug is present in that particular area, and clearance occurring only at liver and kidney. So this is a simple Pbpk model, and it assumes that way. However, if we have to have a Pbpk model for absorption from the gut, then it is complex, because we have to assume that the drug available in the lumen as unabsorbed drug, while the drug available in the enterocyte as absorbed drug. Also, it assumes that the absorption across enterocyte and distribution to different tissues is by passive diffusion only. So mostly the gut compartment model assumes the transfer of drug is by passive diffusion. However, we also have models to incorporate active transport process if required. Okay, so what and all information can be used in Pbpk modeling? For a particular species, we have to have information about the tissue volume, surface area. For example, if you want to know what happens to a drug in liver, we should know the volume of the liver, surface area of the liver. In addition to that, we should know gastric emptying, so that how much time it will be taken for the drug to be absorbed, the intestinal transit times, and pH. So for a particular species, all these parameters may vary with feeding. So this also should be considered while taking all this information. So there are lots of commercial software which considers all this information about various species, and then this can be used to simulate the value in human beings. Okay, so what is this human population physiologically based pharmacokinetic model? It is to find out the physiologically based pharmacokinetics in different types of population or the population of interest. For example, healthy adults of different ethnicities, those with hepatic or renal impairment, smokers, either in young or elderly people, or in obese. So this human population physiologically based pharmacokinetic models can be used to simulate the data in our population of interest. So let me explain some of the studies where this Pbpk model was used. So this is one study where they have used Pbpk model for finding the pharmacokinetics of propofol. So they say that these authors have concluded that a Pbpk model using human parameters and a simple description of tissue binding provides a good description of human propofol kinetics. So let's see how they have found out the kinetics of propofol. The first thing what they did is they have made these compartments. When the drug is given through the vein, what are the tissues where it is distributed, and when it goes to the lung, how much drug will be eliminated through ventilation. All those information has been taken into consideration, and to simulate these information, we need the blood flow, volume of the organ, partition coefficient, and clearance terms. So where do we get all this information? We get it from the literature. For example, the blood flow to various organs in human beings, these authors have taken from Guyton. In mouse, they have taken from an article called Altman. For rat and dog. So in other words, all this information will be taken from the available literature, either standard textbooks or from published articles. Also, the organ volumes will be taken from available literature. So we need the blood flow to the organ, volume of the organ, and then all these things will be simulated to find out how much drug, how the pharmacokinetics of the drug happens. Similarly, one more recently published article, which says physiologically based pharmacokinetic modeling of inhaled nemerilacid. This is, they have slightly gone to the advanced model. So when a drug is inhaled, site of inhaled dose administration will be when the inhaled drug is taken, where and all it gets distributed. So they have made these compartments like extrathoracic, thoracic, bronchiolar, alveolar, lymphatic, systemic, where and stomach. So this also they have considered, like when a inhaled drug reaches the stomach, what happens? So for that, they have used this advanced compartment absorption and transit model, which says when the inhaled drug reaches the stomach, where and all it reaches: stomach, duodenum, jejunum, ileum, cecum, and how long it will remain there depending on the blood supply, from where it will be eliminated, what will be after reaching liver, what happens to it. All these things are taken into consideration in this advanced compartmental absorption and transit model. So let me say, what is this absorption model? An absorption model is when the drug is taken orally, it considers either the drug is transported by diffusion or carrier mediated transport, and the respective values are found out, like Vmax, Km, etc. is found out. Whereas in this advanced compartment absorption transit model, in addition to the absorption, it also considers the various UGT enzymes available in the intestine. Not only the various UGT enzymes, the levels of specific UGT enzymes in different intestinal compartments are used to find out how this drug will be metabolized and what happens to this. And the UGT levels will be again taken from the published literature. Whoever has done work on these different types of UGT enzymes, their expression in small intestine, liver, etc. So all these values will be taken from the previously published literature. So in addition to this, the regulatory authorities like US FDA, EMA, and the regulatory body of Japan, they are also having guidelines for physiologically based pharmacokinetic models. Especially, they recommend physiologically based pharmacokinetic model with respect to drug-drug interaction. So if you look at the uses of Pbpk model, it's purely predictive in nature. Apart from drug development, it can also be used for toxicity assessment. Usually the multi-compartment model with compartments corresponding to defined organs or tissues with interconnections corresponding to blood and lymph flows. And we need to have a priori on anatomical and physiological structure of body to certain extent, as well as should also know the biochemistry of the particular thing. So these models are used mainly for drug delivery system with respect to liposomes, nanoparticles, nanoemulsion, etc. To know the pharmacokinetics. Apart from doing the study in human beings, we can do the physiologically based pharmacokinetics. The information can be obtained from available in silico, in vitro or animal studies, and this can be tried to simulate in humans. Also for large molecules like pegylated protein monoclonal antibodies or antibody drug conjugate, we can use these models to find out the pharmacokinetics in humans based on the available data. Apart from that, the modeling approaches can be used for finding out the safety of cosmetic ingredients. In addition, if we combine the Pbpk with PD model, we can also know the drug response in different species, populations, or disease states. So if we look at the limitations in human Pvpk model, so actually all the values which we get is from different species, and based on extrapolation from animal studies, we find out the value in humans. So the values which are used may be uncertain. The tissue partition of solids has complex dependence on protein and lipid binding. So it can vary from tissue to tissue. The tissue partition, whatever value we obtain, may not be really correct sometimes. So coming to the applications of Pbpk, it is used for understanding the mechanism of a drug. It is useful for predicting doses in human beings through simulation, to predict the drug-drug interactions or steady state level of the drug, assess the effects of influx and efflux transporter, to track the multiple metabolites and what happens to them, also to conduct virtual population studies. So we have been talking so much about models. So what is a good model? A good model is something which is accurate and makes precise predictions, provides information for better understanding of the system. It is rational, consistent, and reasonable. It can be validated. It should be possible to make small changes within the data. It must be a simple procedure. The model should be flexible, and it should be used and completed on time. So just a question for the audience. How a useful model should be? Should it characterize data for a report or publication, or better understand the system under study, make predictions for future studies, or all of the above? So answer is all of the above. The useful model should serve to characterize data for a report or publication, should understand the system under study, should make predictions for future studies. So which is considered as a best model? There can never be a true model. It is just one model is better than the other model. So there is nothing called right model or wrong model. One is better than the other model. So always while choosing the model, we have to choose the simplest model and which is easy and flexible for us to do. That's the carry home message. Thank you.
Thank you, madam. Thank you very much for this wonderful deliberation. So actually, madam, Professor Urmila Thatte had left because of some urgent meeting of PVPI. So on behalf of ISCR and IPS West Bengal, we are really thankful to you, madam, for giving us your time and taking this very complex topic in a very simple way. So this will be a very good learning session for our postgraduate, postdoctoral students. So we are really thankful to you, madam. If any questions, so we can take those questions. If you have any questions, I'm requesting all the delegates, if you have any questions, you can ask in the chat box so that we can address those questions to madam. And otherwise, you can also raise your hand so that we can allow you to unmute and you can raise your question to madam. I think we are recording all to these events, and this will be available in our website. If, with permission from you, madam, is there any issue, madam, if we just download, record it in this
no issue,
and we can upload it in the YouTube? So I think PPTs will be not there, I think. There is no PPT will be shared, but you can, if you want to learn again and again, the video will be there. You can go back to the YouTube link and you can view these videos. This will be available in the website of Indian Pharmacological Society West Bengal chapter, from where you can get this video. So any questions? If you have any questions or any doubts, you can raise your hand. We can unmute you. So I think, madam, there is no more questions in the chat box. And once again, on behalf of Indian Pharmacological Society West Bengal chapter and Indian Society of Clinical Research East Zone, I thank you, madam. And also, we are really thankful to all those faculties who were with us today. And we are also thankful to all the delegates who had participated. There is almost 90 to 95 participants today in this Zoom, and we have another recording that is streamed in the Facebook page. There is also almost 50 to 60 participants who are there. So we are really thankful to all delegates for being with us. And this link you will get again if you want to revisit what our faculties had taught today. You will get it from the YouTube link. And with that, on behalf of Indian Pharmacological Society West Bengal chapter and Indian Society of Clinical Research, I want to thank you all. And also we are really thankful to Inovocare e-Academy for providing us this online platform, to use this online platform. We are really thankful to Inovocare e-Academy. With that, thanks to all the participants, thanks to all the respected faculties who were with us today. Okay, so bye-bye. We are now ending the session.